Hello everybody, this is Dr. P. Hariprasad making this small presentation about Mendelism and monohybrid cross. Well, the main purpose of this presentation is to highlight the real achievements of Mendel. Well, monohybrid cross is the simplest experiment conducted by Gregor John Mendel in the middle of 19th century. And based on the results of this cross, Mendel proposed law of segregation, also known as law of purity of gametes. Well, this is all well known and well explained to the students of +2 all over India. But my purpose in this brief presentation is not to repeat the simple facts about monohybrid cross, but the main idea is to approach monohybrid cross from a different angle, from an angle of Mendel. Now let us briefly state the fundamentals of this monohybrid cross. Mendelism, monohybrid cross, a cross in which two plants are subjected to hybridization differing in one character. And this cross was done by Mendel in garden pea, Pisum sativum. Mendel selected pure tall plants and dwarf plants. Pure tall plant is a tall plant that will give only tall progeny when subjected to repeated self-pollination, which means genetically the tallness is pure. And dwarf, by the way, is always pure. Dwarf cannot be mixed. When crossed, a tall plant and dwarf plant produced F1 progeny, which are all tall. Well, this is where Mendel started his deductive logic analysis. He used a very innovative mathematical method to explain this particular result. Pure tall crossed with dwarf will yield all tall progeny. But this progeny tall are not same as the parental tall. Parental tall are pure. But progeny tall are not pure, because if the progeny tall are subjected to self-pollination, you will get in the progeny both tall and dwarf. The progeny are tall as well as dwarf, F2 progeny, if F1 progeny are subjected to self-pollination. Well, it is a totally different issue that these tall progeny and dwarf progeny appear in 3:1 ratio in F2 generation. That, of course, is the basis for the Mendel's first law of inheritance, that is law of segregation, also known as law of purity of gametes. But my point here is that Mendel reasoned that this progeny tall had a dwarf trait within it, which is not expressed, because this tall plant, if subjected to self-pollination, will produce dwarf progeny. So there is a dwarf factor embedded within the tall plant. There is no such dwarf factor in the parental tall. So this is a pure tall, this is a mixed tall. And he has designated tall trait as capital T, small trait, the dwarf trait as small t. And this designation in letters was described as genotype. So the F1 progeny had a genotype of capital T and small t, indicating that there are two factors present together. Whereas the parental tall has two factors which both are capital T's. And the dwarf plant always had two small t's as genetic factors. Now, why 2 here, 2 here? Because the F1 progeny has 2. Every character is decided by 2 factors. These 2 factors can be similar or dissimilar. If they are similar, it's called homozygous. If they are dissimilar, it's called heterozygous. The F1 are heterozygous, whereas the parentals are homozygous. One is homozygous tall, the other one is homozygous dwarf. But then one of the factors, traits, is expressed in F1, the other present but not expressed. The one which is present but not expressed was called recessive trait. The one which is expressed in F1 progeny was called dominant trait, and that constituted the main principle of dominance. Now, Mendel reasoned that there are 2 factors for each character, and these 2 factors get separated. One of the factors is passed down to the progeny. Of these 2 capital T's, one comes to the progeny. Of these 2 small t's, one comes to the progeny. Therefore, the F1 progeny have capital T and small t, heterozygous genotype. And this was actually called law of segregation or law of purity of gametes. The 2 factors which are together in parents or in progeny must get separated during progeny production. Just as these 2 factors are separated, these 2 factors will also get separated in the next generation during gamete formation. That's what Mendel tried to test. He tried a cross which is F1 into F1. It is sometimes called F1 self-pollination. Now, when F1 are subjected to self-pollination, the progeny are tall as well as dwarf in 3:1 ratio. Mendel made 2 assumptions in making an F1 into F1 cross or F1 self-pollination. One of these assumptions is each F1 parent produces capital T, small t gametes in equal proportion. The second assumption, each gamete has an equal chance of meeting either capital T or small t of the other parent, which means there is no interference of the third party. The formation of gametes is a chance-based event. That is why capital T, small t gametes are produced in exactly equal proportion, and that there is no third party's interference in gametic meeting. Any gamete can meet any other gamete with equal chance. If these 2 assumptions are correct, then we can calculate the result of F1 into F1. Mendel did exactly the same. He has, even before conducting the F1 into F1 cross, calculated mathematically on the basis of these 2 assumptions what could be the possible results. If each F1 parent produces gametes in equal proportions, say 50% capital T, 50% small t, this is the result. These are the gametes of one F1 parent. These 2 are the gametes of another F1 parent. And this gamete can meet this gamete or this gamete with equal chance. There is no preference. There is no interference. There is nothing. It's all chance probability. Any gamete has an equal chance of meeting any other gamete of the other parent. That being so, we can easily calculate what are the outcomes of gametic meetings. If these 2 meet, we get homozygous tall. If these 2 meet, we get heterozygous tall ones. If these 2 meet, we get heterozygous tall once again. And if these 2 meet, we get a recessive dwarf progeny. Now, the ratio in which these progeny will appear is 3:1, 3 tall and 1 dwarf. If you consider genotypic ratio, it will be 1:2:1 ratio, which means 1 homozygous tall, 2 heterozygous tall, and 1 dwarf. This actually means if you have 4 progeny, 1 is likely to be homozygous tall. Similarly, if you have 4 progeny, 2 are likely to be heterozygous tall and 1 likely to be dwarf progeny. This is called genotypic ratio. This is called phenotypic ratio. Monohybrid Mendelian phenotypic ratio is 3:1. Monohybrid Mendelian genotypic ratio is 1:1. If these two assumptions are correct, then we should get a phenotypic ratio of 3:1 or a genotypic ratio of 1:1. Remember, only if these two assumptions are true. In other words, if these assumptions are not correct, we would not get these ratios. That is the mathematical logic employed by Gregor John Mendel. Now, having calculated the possible result on the basis of two basic assumptions, which are chance-based assumptions, Mendel then went on to actually do the cross F1 into F1 or F1 selfing. When he crossed F1 into F1, keeping in mind that it will be 3:1 phenotypic ratio or 1:2:1 genotypic ratio if these two are true. Then, as expected, Mendel crossed F1 into F1 and he ended up with 3:1 ratio. This actually means that in inheritance, gamete formation is chance-based and gametic meeting is also chance-based. He would not have got 3:1 ratio if these two assumptions or any of these two assumptions is wrong. He got 3:1 ratio, therefore it is confirmed that these two assumptions are right. Which means Mendel actually calculated and arrived at these ratios, mathematical ratios, much before he actually undertook the F1 into F1 cross. And his mathematical expectation came right, and Mendel therefore concluded that in F2 progeny, tall and dwarf progeny appear in approximately 3:1 ratio. And this ratio was used to propound the first important law of inheritance, that is law of segregation of law or law of purity of gametes. It states that the two factors which are together in a F1 parent, F1 progeny, are separated, and each of these factors enters a separate gamete. Both these factors do not enter the same gamete. So there is segregation, separation of these two factors during gamete formation. Remember, these two factors are brought together in sexual reproduction when gametes fuse. Therefore, Mendel finally established that there is no third party's interference in inheritance. Inheritance of factors is a chance-based event. It is a probabilistic event. There is no interference of any factor, third party in inheritance. That, I think, is the most important conclusion of Mendelism. We could discuss the inheritance of a pair of factors or two pairs of factors. The second law also proves this. Inheritance in organisms in general is a chance-based probabilistic event. Now, this monohybrid cross is often taught as if Mendel was surprised with 3:1 ratio, as if Mendel got 3:1 ratio, he stumbled upon accidentally this 3:1 ratio. This is, however, not true. And Mendel actually has mathematically expected 3:1 ratio and got this 3:1 ratio. The implication is very, very far reaching. It means that inheritance is chance-based. Inheritance is probabilistic. For that matter, all the ratios in genetics that we use, 1:2:1 ratio, 3:1 ratio, or monohybrid test cross ratio, 1:1, dihybrid ratio 9:3:3:1, dihybrid genotypic ratio 1:2:2:4, 1 to 1 to 1. All these ratios mean only one thing, that inheritance is a chance-based event. Inheritance is a probabilistic event. That is the single most important contribution of Mendel to the understanding of heredity and inheritance. Thank you.